Hello students, welcome to Eduauraa. I am Prajakta Palande and I am your biology faculty. In today's session, we'll go ahead with the chapter Biotechnology, Principles and Processes. Can you suggest an enzyme that will cut this DNA? I've taught you these enzymes. They're called as restriction endonucleases. So, we will use restriction endonucleases here, and that human DNA will be cut into fragments. And these fragments will be screened. What do we mean by screening? Screening means to check, because we just need the insulin gene. So these fragments will be screened for the desirable gene. And like I have already told you, the desirable gene for this particular experiment right now is the insulin gene. So the screening was done, and this is the insulin gene that we are interested in. So we have the desirable gene now. Let's keep this away for some time. Let us now focus on the bacteria E. coli. E. coli stands for Escherichia coli. E. coli has its main bacterial genome, that is here. But other than the bacterial genome, it has this extra chromosomal circular piece of DNA, which is called as the plasmid. So I have an E. coli. There is a plasmid in this E. coli. We will remove this plasmid out. Here. This is a plasmid. Now we will use the same restriction endonuclease that I used for this human DNA. I will use the same restriction endonuclease on this plasmid as well. So this plasmid gets cut. And this is what I will have left now, because the restriction endonuclease cut the plasmid. Now do you see there is a space in the plasmid? This is where I will add that insulin gene. But do you think that insulin gene will just get joined to that plasmid? No. I will need an enzyme that will join these two pieces of DNA. Can you suggest an enzyme? It is called DNA ligase. DNA ligase helps in joining the pieces of DNA. So now, the human insulin gene has been joined to the plasmid, which is the bacterial DNA. And this piece of DNA, which is hence formed, is the recombinant DNA. It is also called as rDNA. The R stands for recombinant. Why recombinant? Because there is a part from human genome and there is part from bacterial genome. So it is two different DNAs combining with each other. It is called as recombinant DNA. So this is the recombinant DNA, which is also called as rDNA, or it can also be called as chimeric DNA. What will happen to this recombinant DNA? We are going to put this recombinant DNA back into where it came from. So where did that plasmid come from? It came from the E. coli. So that is where we'll put that recombinant DNA again. This recombinant DNA will be introduced back into that bacterial cell. And now this bacterial cell will multiply. And when it will multiply, it will also multiply that insulin gene, because it now thinks that the insulin gene is a part of its own plasmid. With that, let us end today's session. This is Prajakta Palande signing off. You keep watching Eduauraa, and I'll see you in the next session.